let's then start. I will, as Pascal said, I'm a statistical physicist. The audio. So, to not get distracted. Great. So, I'm a statistical physicist, and my interest is applications of statistical physics to biology. So the idea here is, yes, thanks, to use tools from statistical mechanics to understand systems, biological systems, and also to use inspiration from biology to get new interest in physics. So we build the models in order to understand biology, and these models will give us also some new information about physics. So in general, I'm interested into non-equilibrium statistical physics, applications to neuroscience, and then some stuff in evolutionary biology that I will not touch today. Okay, so the main topic is going to be like criticality in the brain and dynamical systems in the brain. In order to introduce the topic, I will first talk a bit about criticality, because maybe not everyone here knows what is criticality. So the concept is very simple. We are very used to see water, for example, in different phases, like solid, liquid, or gas. And we know that depending on the conditions, for example, temperature, we are able to see water in these different phases. So there is a control parameter that allows us to say what is the phase that we have. And the difference between these two phases is basically the ordering. In the solid phase, the atoms are very well packed, are ordered, while in the liquid phase, the atoms are very, very disordered, okay? So this does not apply only for these three typical phases of matter that all of us are very used to, but also, for example, to magnetic systems. If we have a magnetic system that can have a small magnets with up and down poles, we see more or less the same thing. We have a phase in which everybody has to be more or less pointing upwards or downwards, or a phase in which the thing is completely disordered, and this depends on temperature or the noise fluctuations that the system may have. So this happens too in other kind of systems. For example, if we have something like a spreading dynamics, spreading dynamics is something that I mean by I'm, for example, infected or active, and I can transmit my activity to everybody else in the network. In this case, there could be a phase transition in which I have constant amount of activity, or I could just die. In this example that I show here, what we do is infecting someone in the middle of a one-dimensional ring, and then see how the activity propagates through time. So you see if the coupling between different neighbors, the contact between them is not strong enough, we have there in blue, then it will die at some point. If it is too large, then it will get an entire fraction of the network and it will spread completely. So in this case, we represent this kind of phenomena with a phase diagram like this, in which the blue part will say it's an absorbing phase. It will go up at a point in which there is nobody active. And then we have an active phase in which we have a constant number of people who is active, right? In the middle, there is this critical point in which we have some very special properties. One of them is that we have scale-free avalanches. This is one of the main markers, okay, of criticality, and it is characterized because if we do this experiment of I just infect one person and I see how it spreads, the statistics of this event are scale-free in the sense that I could infect just 10 people, or I could infect million of them if the system size allows it. There is no typical scale on how many people I will infect, okay? If you go to any of the other two phases, these things have a typical scale. Like in the blue one, it's like, oh, in average, you will infect 10 people or 100 people before dying. This does not happen in the critical point. At the end, the critical point always will go to zero, but the time that it will take to do so can be as long as you want. Okay, and the interesting thing is that this has been seen in the brain. So the first paper that observed this thing was Bex and Pless in the Journal of Neuroscience, 2003. So they were measuring LFP activity in cortical slices, and from this kind of experiments, they were able to find this kind of avalanches. And once you have this, then you might say, okay, if we have the avalanches, it might be that the brain is following this kind of process in which neurons are trying to spread activity and are spreading activity in some kind of marginal way between the two phases. The problem is that it is not so easy. Like criticality will imply this scale-free phenomena, but the contrary is not automatically true. So it will be more complicated. So you might question why this criticality is important. The first thing I want to say is that there is different kinds of phase transitions. Before some of the physicists in the audience kill me because I started comparing like phases of water with the magnetic ones, and they are not the same thing. If you go to the phase diagram of the water, we have here liquid, we have the vapor. You see all these lines are phase transitions between the phases of matter, but they are not critical. The only critical point here is marked in red, okay? So there are different kind of phase transitions. These kind of lines are like first order. You remember that the graph I showed before was some kind of continuous line from the two states. Here is two discontinuous lines, and you jump between the states. So this is something that can happen. And this, in theory, they are not critical. So as a statistical physicist, our idea for the brain, or one of the ideas that we have for the brain, is that it would be nice if we can get this kind of phase diagram, like some parameters and some dynamical regimes or phases, and how are the transitions between them, so we can understand this map where we are, right? So as I said, we are interested in criticality for a reason, and it is because critical points have special properties, and some of them are very interesting from the point of view of biology. For example, we have large susceptibility, so you will react very strongly to a kind of external inputs. The correlations between the systems are very long range, so information can flow in a very easy way. You have lots of dynamic range, so all of this is very good. And it has been like very strong evidence for other biological systems that we might be in this kind of point. For example, for flocking of birds, and we have also chemical transmission of information in bacterial communities, and then for genetic networks. And there are more things that will appear in a second, like we have also ecology. We have, first school and swarms. And I would like to recommend a review from my ex-PhD supervisor, Miguel Ángel Muñoz, that has some kind of compilation on what is the evidence for all these kind of systems right now. So as I say, we want to have this kind of map for the brain. The brain has like many different dynamical states. You have asynchronous spiking, you have oscillations. This graph is for organoids, but in the brain also we have like real oscillations. And then we have, I said, also these avalanches. So there are many things that we want to put all of them together, like in this water phase diagram that we had before. So in this case, I will gonna focus on the oscillations part and the transitions between oscillatory regimes and other kind of regimes. I will start from the very beginning, which is the Millfield-Kuramoto model. And from this, I will start adding different elements, and we will compare which are the differences and which are the differences and similarities. So the Kuramoto model is a very simple model in which we have a phase. So you are in some part of a cycle, and you are able to move there using just two ingredients. One is your own frequency. So it's like some kind of sinusoidal thing that makes you go around the cycle. And then there is this pink term, which is interaction between different oscillators. So we put many oscillators there, and this interaction term might look complicated, but the only thing that it does is try to attract all the oscillators so they want to go together. So the thing is, all oscillators interact with everybody, and the idea is that if the coupling J is large enough, we might be able to put everybody together, even when they have different frequencies. So the frequencies we obtain from any probability distribution we want. One of the most popular is the Lorentzian or Cauchy distribution instead of a Gaussian, just because this one has better properties if you want to do the computations by hand, but you can use whatever you want. And then we have the Kuramoto order parameter, okay? So the way in which we measure order here is like if the oscillators are very packed. So we do this by computing this complex number and taking the absolute value. If this absolute value is close to 1, then all oscillators are going together. If it is close to 0, they are perfectly spread. This check that this does not have any stochastic noise. There is a version also in which we can include the stochastic noise, but for the sake of simplicity, I will start talking about the typical Kuramoto model, which has no noise. Okay, so this is how the model looks like. It has this kind of continuous phase transition. And in the left, you can see the ones oscillators that get synchronized, and the other one is when they are not synchronous. So what you see is that in the left one, after some point, there is this big cluster that forms and travels all together. So this is something that is more or less synchronized, and in the other one, it's just things that are distributed randomly through the cycle. So this is more or less the behavior of the model. And it depends, as I said, on how much your coupling is and how different the frequencies are. So the way that we usually like to work with this model is that, okay, this very big sum, it's a bit ugly. So it is possible to change it into this simpler form, just introducing this R, which is the absolute value of this complex number I was talking before, and the collective phase of this whole thing that is moving, okay? So it is the same equation. I just did some small black magic to it. I have details on how I do all this kind of stuff if someone is interested at the end. And what we do is to study the thermodynamic limit. The thermodynamic limit is a very fancy physics way to say I will put as many oscillators as I can. So what we do usually is we study the density of oscillators. So we say, okay, we take a small fraction of the circle, a small arc, and say how many people is inside of here. And we study for each arc length, how many people is going to be here. This is the equation that governs that kind of density. Is called the continuity equation. It is very easy to understand. For example, if there is a Spanish comedian that says that the chickens that go in must equal the chickens that go out, and this is exactly what this equation does. Okay, so it's how the density in the arc changes, and this is how many chickens are going in and how many are going out. So if you have more going out than going in, the density increases. It's just that. Okay, so the idea is, the problem is that once you start making all this in the continuum, working with densities, this relatively simple shape of the ordered parameter becomes this pair of not so beautiful integrals, and things become a bit more complicated. So this prevents the model to be solved in an easy way. And it has also, for noise, it's even worse because there is this diffusion thing here. We will not enter into that. And then there is to solve it, we have the Odd-Antonsen theory that you see. It was proposed in 2008 when the Kuramoto model was way, way older, from I think 70s, 80s, something like that. So this is, I will not do the computations. I can show them to you at the end if you want. But basically you take, this is very small abstract on how to do that. You expand densities of Fourier series. You see that these coefficients are like suspiciously similar to these parameters, complex parameters that we have. And then you assume that I want to simplify everything. So I will not have many terms in the series. I will only have one, and all the others depend on this one. And with this, then you find an equation for this thing. So you see that we started from an equation that has information about all the oscillators. It's a microscopic equation, while this is an equation for this complex order parameter. It's a collective equation that governs the collective dynamics, what everybody is doing, seen as a whole. Okay. So if you look at this equation, of course, you find this line. This is actually how I draw it. And this kind of equation is used, for example, in many papers yes, you couple some of them, or you analyze this thing, and you can use it for to study brain dynamics. And another thing to visualize this is that you could add some kind of raster plot to the oscillators. So every time we complete a phase, then you will put a point, and then you are able to see that the synchronous one looks some kind of regular oscillations, and then there is some noise going on in the synchronous one. Okay, and in the synchronization transition, there is some kind of hidden structure there that is not very clear. So this is the mean field thing. we have oscillators that are connected all to all. This is not extremely interesting for the case of the brain, in which we usually have networks. So the next thing I'm gonna do is to add some topological structure to this model and see how the structure changes the results. Okay? So one of the first things that appear is that if I want to study this thing in low-dimensional systems, the thing becomes spicy. There is this Mermin-Wagner theorem that says that continuous symmetries cannot be broken at finite temperature in systems with dimension less or equal than 2. This is, and again, a very fancy physicist way to say this kind of oscillators cannot be synchronized once you put them in a lattice. What you will see is this kind of behavior in which vortices appear and they go around all the time. Okay? So actually describing these vortices and how they appear and so on, it's Nobel Prize 2016, they related to the Koster-Lithaules transition, blah, 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 blah. But the thing is that in low-dimensional systems, if you try to synchronize these oscillators, you will fail. And I remark this because it will become important later. Okay, so we have more things. For example, what happens if we simulate this model, not in an Erdos range, boring network, but something that has escape-free connectivity, because the brain, we know it has some kind of escape-free connectivity. Then again, you see the results, results are very recent. This is 2019 to have a clear answer to what happens to this, which is basically that the oscillators will always synchronize, maybe not too much, but they will always synchronize in this kind of networks. What you see here is the location of the critical point as I increase the size of this Erdos range network. And what you see and what these people predict with theory is also that the critical point will go to 0, meaning that no matter which coupling you have, you will end up synchronized. So having this kind of scale-free network will help and promote synchronization. Let's even add some more ingredients to the thing. So we know also that brain networks are kind of fractal and hierarchical-modular. So you have different modules that are connected in layers. And this has been shown to produce Griffith phases, which I will explain in a second. So how are these hierarchical-modular networks? It's simple approaches. I take some brain regions or oscillators or whatever, and then I start connecting them by pairs. And then the next thing I will do is just to say, okay, I will just connect these modules together with a weaker coupling, and I will continue to do so as I go up. So I have very well connected modules that are less and less connected as I go up in the hierarchy, but there is a whole hierarchy of connected modules. So when what happens with this is that this phase transition that we had before starts to go to move with the number of layers. So it's very easy to synchronize these people here in the cyan, but it's not very easy to synchronize all the modules together when in the yellow one. It gets synchronized by layer, little by little, as you increase the coupling. And in the thing is that this can give rise to these Griffith phases. So the thing is that let's combine all these three stuff. We have scale-free networks, we have the hierarchical modular ones, and we have that the oscillators cannot be synchronized very well in low dimensions. The problem is that if you estimate the dimensionality of brain connectome is between 2 and 3. So oscillators will not like to synchronize very well in here. But they want to do so because also connectivity is kind of scale-free. The result is that we have this kind of Griffith phase. So you see all this region, instead of having our super nice phase diagram as before, it's some kind of noisy stuff in the middle. And the whole region has properties that are similar to critical systems. So instead of having a single point, we will have now an extended region with all the nice properties. And the idea of these people here, which are most of them are from my lab at Granada, is that, well, it might be even better in this case because I don't have to fine-tune my dynamics to this critical point. I could just live in an entire region of critical-like properties. Okay?
Can I ask a question?
Yes, of course.
So what does the dimensionality mean in the context of like brain connections and networks? So
Yeah, in this case, it was also related to what we were talking about yesterday. In this case, this D, I believe that it corresponds to a spectral dimension. So you get all the eigenvalues of the metric, you put them together, and the slope, you rank them from larger to smaller, and the slope of the thing is what will give you this stuff. However, this is also what we were discussing a bit. And the other is that there are many ways to define what a dimension is. And depending on your model and what you do, one definition or other will be more adequate. According to specialities Millán, in this case for oscillators, the spectral dimension is what you should be looking at. But of course, there could be more things. Yes, please.
Yeah. So when you say this is scale-free, more or less, the way I interpret that is to mean that the extent of a region that has a local base, it doesn't have a typical size.
This is a good question. In this case, the scale-free appears in the equations that you look for the order parameter, right? So you have the oscillators. We do this limit that I did before using the densities and so on, and you get this equation for the complex order parameters. So the scale freeness will go in there. What is scale free is the Kuramoto order parameter. So you will find that the system is mostly asynchronous, and then it has some fluctuations that makes it very synchronous and then go down. So and these fluctuations, it's
So
what
across
would be
time rather than space.
No, it should also happen in a space. The problem is that you cannot visualize this happening in a lattice, for example, because in a lattice it will not happen. You need to go to, for example, a 4-dimensional space in order to be able to measure like correlations from point to point, and then having this correlation, but it will be correlations on the order parameter, not the correlations in the phases. I don't know what they will do. One will have to check. Yes.
Just to continue this, and this dimension being between 2 and 3 is for some specific connectome, for some specific species, or did this person look at everything that's out there?
This is for, this is for human data. I guess that the other connectomes should have different dimensions. However, given how the brain grows in a 3-dimensional space, having a dimension larger than 3, it could depend on. It's relatively complicated, right? Because things live in a physical space. So you can have the network connectivity, of course, can go over this dimension, but the most intuitive thing seems to be that the way that you grow is constrained by growing in a Euclidean space that is subject to these rules, and it has a dimension. Yes. Okay. So other oscillator models. So for now, we have been including more complexity into the topological part, and we have been leaving the Kuramoto model, let's say, we have been leaving the Kuramoto model fixed. We want to see now what happens when we include other dynamics. So one of popular options is this Kuramoto-Sakaguchi model. The only difference with the usual Kuramoto model is that this small alpha inside of the coupling, that it's intended to mimic time delays. So you have 2 oscillators, and they want to get synchronized with a certain dephase, which mimics that the signal from one oscillator to the other will need some time to arrive. And this is, of course, very useful when we are talking about brain dynamics. So one particular feature of this model that is the same. And still, if you simulate this model, for example, in 1 dimension, you will have that half of the oscillators do synchronize and half of the oscillators do not, which is a relatively weird behavior. Okay, so this kind of time delay are able to produce some structure that is different. One of the things I do not like for Kuramoto model is that all oscillations are linear, and I want to include excitability, which is present in neuronal systems. So what you see here is that the oscillators now tend to get cluttered in the down part, and then just they just jump from time to time going back, right? This is because this sine term here adds some kind of a structure, adds another transition depending on the frequency and the excitability. And the idea is that this gives more freedom to our oscillators because instead of just oscillating, they can also get stacked if you want, depending on how you parameterize them. So, are you responsive? So we use this, and I use this Oden-Thomsen theory in order to construct entire phase diagram for this kind of active rotators, which is more complicated than the phase diagram for the Kuramoto model, which is just a line. This is more similar to the thing that we saw with the phase diagram of water, just because it's more complex because we have more parameters. And I did analyze all the dynamics on different bifurcations. We have this type 2 and type 1 bifurcation. This is different kind of phase transitions. And then there are some points, as in the water diagram, which are special points. And what we saw is that in one of these special points, we have a dynamics here that is not trivial at all. All the other ones are like just oscillating, or you get stuck, or something relatively stupid. But that special point in the middle has this very nice raster plots that are similar to what you will see in some neuronal systems. And they are able to produce these scale-free avalanches, similar to the ones that Bex and Plens were obtaining. So it could be like one possible explanation or generative mechanism in order to get these avalanches to be to this kind of point. So we are able to get this scale-free behavior from the oscillatory models. Okay. I will like to finish just putting everything a bit together. We have been talking about topology. I have added some stuff about dynamics. There is way more. There's a lot of literature in oscillator models, and it is very easy to change this small detail or this one and then do an analysis. So there is really a lot of things. But I don't have time, of course, to go through all. So the idea is that people have been assuming that, oh, we have this very nice equation that comes from the mean field. So let's put that equation to one brain region. The brain region will be composed by smaller things. So we get that over the brain region. And then people just check the functional connectivity, and they compare with simulations of this model, and you can see that then there is some of the regions when you do, for example, parameter inference, that are close to this critical to this critical point. No, so for example, this is from this DECON 2017, and they say these blue regions here will correspond to regions that are close to a critical state. Okay. Then there is a discussion on why these regions and not the others, and what functional role is a dynamical state has, and so on and so on and so on. Okay, so in general, all this goes like you can have a model and you do a phase reduction. You can do this with many models. So this phase reduction will usually look like this in general, like Kuramoto model with this Kuramoto-Sakaguchi term, which accounts for the delays between different regions. We do this Oden-Thomsen theory in order to have a better equation for each one of the regions. And then we combine it with structural connectivity data in order to compute observables like functional connectivity, yes, correlations between different regions, synchronization between them, this kind of stuff, right? And there is a whole literature of people doing this during the last years. And one of the things, for example, is that you start from the Wilson-Cowan model, you do the whole thing, you have this frustrated synchronization that corresponds also to these Griffith phases I was saying before. So we also did some kind of stuff regarding this, which is like we take different topologies, we take this phase diagram with different dynamics, and then we combine them, and we check like what happens when you combine each kind of dynamics, which is kind of topology, and assess what is the dynamical richness using this Fano factor. So the wider the Fano factor distribution is, the more dynamical rich the thing is, that's what some people have measured. So we repeated this kind of stuff. And so that the more complex networks, for example, with this HMCP has hierarchical modular structure and also scale freeness, and they usually tend to give better result, especially for the critical state. Okay. And the last project I'm doing is I'm taking the fMRI data from the Human Connectome project, and I'm trying to do inference myself using some of the new techniques that people is developing in Tübingen in order to try to get an idea of the probability of observing certain parameters. No, so for each brain region, I try to see what are the parameters that correspond to it, and then what is the dynamical state of that region. And then from that, I will try to see which regions are closer to criticality or not, and why. And this is more or less the general idea I'm working on right now. So, given all that, I think it's time for me to thanks to all the wonderful collaborators, the people I work in my PhD and now in the Levina lab, and to you all for listening to my talk. And yeah, if you want to talk to me, ask questions, whatever, I really like to talk. In case you have not noticed, this is just reach to me, and I would like to announce this is maybe a slightly secret, but it's, we have been accepted for a workshop on the Bernstein conference on a whole brain model synchronization. So if you are interested in these topics, come with us on Berlin, September this year. Thank you so much.
Is there any question or more question for Victor?
Yes.
Thank you for your talk. This may be a silly question because I'm not too familiar with this particular thing that you're talking about. I got at the beginning where you had the phase transition of temperature and pressure. And then when you move to the brain, you had this alpha and sigma. And I assume one of them is some sort of energy or what you're feeding into the system.
Yeah, in this case, let me put it again.
Yeah, like here. Yeah, this is
so. Let me also show it here. We have, this is the model in which, in this case, we have a stochastic noise. So what we are using for the phase diagram is the intensity of the noise and how much excitable is the system. So here we have several parameters. We have also the omega and J. So if you want to have the entire mapping on which phase you are, you will need a four-dimensional phase diagram. But what I do is basically just fixing two of the parameters, and then I make a cut. And once you check that more or less, there is no new phases once you move on other directions, right? So it's more or less representative.
Okay, but this A is the same for all the neurons in the system.
In this case, yes.
Oh.
Yeah, this is the problem, you know, like, and for example, in the inference stuff I'm doing right now, you cannot assume that all regions will have the same excitability. So this poses an additional problem. If I start like, oh, each oscillator also here, everybody has the same frequency. If I start adding disorder to all of the parameters here, the phase diagram will most probably change. How? I don't know. That's a hard problem. Thank you. Yep.
And you were mentioning phase reduction techniques. Yes. So as far as I know, these techniques don't usually work if you have like strong non-equilibrium systems where you have like a strong coupling between the amplitude and the phase of oscillations.
Yep.
Like, have people been doing these things with, like, I don't know, oscillators, nonlinear oscillators rather than phase oscillators?
No, they have been cheating, basically. So the thing is that, for example, for the Wilson-Cowan model, I think they were studying it in a regime in which it was close to a Hopf bifurcation. So in this kind of Hopf bifurcation, all the oscillations are very circular. These phase reductions, and as he was saying, only work if the oscillators are circular. If they are like ellipses or whatever, you cannot do them because it's not possible to write a single equation for the phase. You need to take into account how much is the amplitude. You cannot just reduce it to one. So this is true. When people do this kind of oscillatory reductions, it's because they assume we forget completely about the amplitude, and this might not be correct. But this depends also on which kind of regime you're interested to study, and so on and so on. Yes.
Am I to understand each node in your network that has this oscillation frequency and excitability and so on as some sort of a neuron or a little sub area of the brain?
Yeah.
And then my next question, I mean, in real brains, they're not just doing one oscillation, and want to do that, they are having several oscillations going on at the same time, right?
Can
You have nested theta, you have nested gamma and theta, and then you have some other rhythm on top, and then you have something that is just like a very ephemeral low frequency bump or so. So, yeah. Would you want to integrate this, or, I mean, if you claim you make a whole brain model?
Yes. So the idea here in general is that you have this kind of model that represents the dynamics of a single area. Let's say that you have the area in isolation, right? So it's not connected to anything. So this area will have some dynamic properties that are intrinsic to it. Of course, when you connect it to other areas, even if I just said, oh, this area has a single frequency here, just from the fact that it's receiving inputs from other places, it will modulate the frequencies, and then you will have like different nested oscillations, for example. So I don't have any graph here, but this kind of nested oscillations can really be seen if you look at individual nodes inside of the network. They emerge as a result of connecting everybody together. So for me, it's not like necessary, let's say, to introduce into my model, oh, I have like alpha, beta, gamma oscillations, and I put all of them inside of the model explicitly, because in the best case, we hope this will be a consequence also of communication between the different nodes. But it is true that maybe this is not enough, and we will need to set like some kind of different models that intrinsically have different frequencies. But this is too easy because it's a difficult problem. I cannot get a brain area, isolate it, and see what it does in isolation. This is not possible to be done. So that's why also doing this inference of parameters is a complicated task.
Could you expand on what you mean by I think you called it a chimera?
Yes.
When you have time delays and how that arises and what that actually means.
So these chimeral states are relatively funny. They are not easy to obtain. They are relatively unstable, but there is the idea that they could be also seen in neural data for other setups. But this is, to my knowledge, this is the simplest one that gives these chimeras. They are called like this just because in this case, all oscillators are the same. And so if you put them in a line, you will expect either all of them to go at the same time or just not to be able to synchronize. No? And what you see is that there is like half of the population gets synchronous and the other half is not. And it is also follows on a spatial structure. So you see there is like half here, all of them are going together and the other ones are not. So for the brain, this could mean that you have some kind of emergent property that will allow you to synchronize or communicate different areas while leaving others to be independent, even when all the areas are very similar to each other. So you will not need an heterogeneity in the parameters in order ways to, just have half of the system asynchronous and half of the system synchronous. And I think this is the main interest on what these chimeras mean. Yeah.
So the fact that you have like half of the nodes synchronizing and half not, is this sort of emerging from the fact both that you have a time delay and that you have variable weights between the connections, these AIJs?
But in this case, these AIJs are just set to have a single line.
Okay.
So it's all of them will have the same weight.
Okay.
And they will be positioned into line. So this AIJ will be just.
And so remind me, the points are on a line, not a circle in this example.
Well, it's a circle because it's you connect the two extremes of the line for periodic boundary conditions, right? Yes, it's a one-dimensional thing. So yeah, it's surprising because of that. There is no heterogeneity in frequencies. There is no heterogeneity in weights. Everybody is exactly the same thing, and still the structure appears.
Cool. Is there time for?
Yeah.
In all these models, you have a coupling that is basically one harmonic, right? It's just sign.
Yes.
And that's why you have this low dimensional description, which is the Otto-Ansonsen ansatz. But if you have two, like you have the first one and then the second one, this low dimensional prescription doesn't work, right?
I think that even if you had like higher harmonics, the Odd-Antonsen solution, I think it will still be, most probably, I think it will still be working. But yes, it's true that this is a very simple shape of the interaction dynamics. So one could consider another more complicated behavior. But yeah, it depends. So the thing is, when you want to apply these models, sometimes it's not worth to put a lot of detail into it because it's already like there are many details in the experimental data that how to model. But I think Odd-Antonsen ansatz will work even if you have like higher. Yes, they do. Yes, because there is these theta neurons that have the interaction is 1 minus cosine to the n, and you can even make n to the infinite, and you can find an exact solution to these guys using the Odd-Antonsen ansatz. Is the Odd-Antonsen ansatz only phase if there is a stochastic noise? Then it is not exact. But for deterministic oscillators, it is hypothesized to be exact in every case.
In this case, would the single neurons be migrating between the two cases, or would they stay in one of them? And if they migrate, what would be the sort of time scale of staying in one?
That's a good question. In this case, I believe that for this model, they stay the way they are. But some other models, when you start playing around with the chimeras, you can find chimeras in time. So people will get stuck in some state, and after some time, they will just change to other things. So it is possible to have a pair of states and oscillators to move between them. I think not in this case.
In that case, if you give a perturbation, maybe you can jump.
Yes. In this case, even for this one, I believe that with a perturbation, it should be possible. But to be fair, it's difficult to set up systems to do this. It's not something very obvious to do. So that's also, there is also the discussion how to generate these chimeras in a very stable, reliable way.
Okay, so with this we can thank Victor again.